towards, now depending on how you want to look at it, towards becoming a larva, an embryo of some sort, or an adult. Sometimes one even comes across the phrase genetic program. So development is thought of as the playing out, unfolding as it were, of a genetic program. There is a similar point of view which has taken hold of evolutionary biology, which is that evolution is to be understood in terms of changes in the genetic composition of populations. So far so good, but changes in the genetic composition which have come about as the result of forces, primarily natural selection, forces which have acted on random changes in gene frequencies arising from the acquisition of new genes or new gene function, or sometimes also the loss of genes, which you can think of as a negative acquisition. So, in any case, both of these pictures place genes at the center and have some kind of deterministic working out of the effects of gene activity, in one case in the course of development, in the second case in the course of phylogeny, as the central explanatory principle underlying whatever it is that we are studying. Now, already in the talks that we have heard in this room today, we have seen that there are possibly other sorts of explanations plausible. Indeed, one of them being trying to look at cells per se, or moving up the hierarchy, organisms per se, sometimes perhaps even groups of organisms, as the basic units on whom change occurs and is later translated into what we think of as a reproducible developmental pattern, or with hindsight as an evolutionary sequence. My aim will be partly to reinforce what the previous speakers have said and to get across to you the notion that this way of looking at things does make sense, and partly also to try and raise the question in your mind of whether genetic change has to necessarily precede form change, or if you wish, organismal change, or it could in certain circumstances follow such change in a manner that we will try and look at in one or two cases. For this talk, I will confine myself to microorganisms, and in particular to one type of microorganism that you have already seen. In fact, you have seen it in action more or less in the talk of Professor Bhaskar. This is this thing called the cellular slime mold, or dictyostelium plus its relatives. As you will see, when we study these dictyostelid amoebae, as they are called, or the cellular slime molds, you will see that there are things that happen which seem to transcend simple-minded genetic explanations, and which seem to demand, or at least necessitate the kinds of explanations that I alluded to earlier. And in particular, these explanations seem to hold good for group traits, collective traits, and these of course are precisely the kinds of traits that lie at the heart of development. I am speaking of development, thinking of development here as a multicellular phenomenon which requires the collaboration or cooperation of large numbers of cells. As I will try to point out to you, not in detail, as I will try to point out to you, there are rather nice parallels between the way one looks at group behavior in multicellularity and the way one looks at group behavior in terms of groups of organisms, what we usually think of as social behavior, so that you can look at social behavior as commonly understood as a form of multicellular development, and indeed multicellular development turning it the other way around as a form of social behavior. In fact, one of the amusing things about some of these ideas is how old they are and how they were thrown up at one time, looked at with interest, and then fell out of fashion as this dominant view that I mentioned earlier took hold. So, we will look at organisms that are facultatively multicellular, which is to say not obligatorily multicellular. Therefore, I will say right away that we are not looking at anything like the human embryo or the fly embryo, I should say human beings or flies or fishes or frogs, all of which have to be multicellular, they have no option. Rather, we will look at organisms which have the option of being multicellular, and the advantage of that, of course, is now you can ask what it is that they need to do in order to become multicellular, or what properties do they need to exhibit in order to show these multicellularities. Now, it turns out that this form of multicellularity, what I call facultative or optional multicellularity, is actually ubiquitous. This particular chart shows what are called the major, and these are sometimes called super groups of the eukaryotic tree of life. The tree of life, which includes creatures like us, excludes creatures without a nucleus. And among these super groups, the kind of multicellularity that I want to bring before you is found in the ophistoconts, which includes us somewhere, found in the amoebozoa, second super group, which includes Dictyostelium that we will talk about, includes straminopiles, alveolates, which are slightly different but also come under this head, Rhizaria. Interestingly, not the super group which has given rise to today's higher plants. This for higher plants themselves, of course, are multicellular, but this form of multicellularity, facultative, aggregative, as you will see, multicellularity, is not found in them. Nonetheless, the point of interest here is that all of these super groups trace back to a presumed putative common ancestor. The dating here isn't terribly good, but a common ancestor who lived possibly 650 million years ago. And we are reasonably sure that these various forms of multicellularity arose independently in each of the lineages which have given rise to these creatures of today. Same kind of thing, but in a different way of representation, which show you that these cellular slime molds, and in particular Dictyostelium discoideum, came after the plant line had separated from the main eukaryotic tree of life, but before the separation of the fungi or the animals. Now, some similarities in a formal sense between these explanatory ideas that I referred to across these supergroups. And the best way of seeing what these similarities are is to look at life cycles. So, here you see sketches of 4 different kinds of creatures, all of which of course are around us here today, but which come from very different supergroups. So, they have lineages which do not share a common ancestor until you go back to that single cell that we thought about. This is the same supergroup as Dictyostelium, amoebozoa. Then you have the excavates, and then you have the ophistoconts, which happens to include us, as I said. And without going through these pictures in detail, you can see immediately that in all of these cases, you have cells which look amoeboid, shapeless in some sense, move around, feed, usually on anything smaller than themselves, probably bacteria. Most of them are found in the soil, so soil bacteria, grow, divide by mitosis after feeding. Many of them have again a facultatively sexual cycle, which we will not get occupied with in the course of this talk. But they go through a phase of feeding, growth, and cell division, therefore increase in numbers. At some stage they exhaust the food supply, and when that happens, as you can see from these sketches, they come together. Having come together, you see the second point of convergence, all of them build this multicellular structure called the fruiting body, which you again saw in Bhaskar's pictures. And this fruiting body has 1, 2 or more cell types, not too many, perhaps 3 at the outside, maybe 4 in some cases. The shape of the fruiting body is common across all of these supergroups. There is something like a stalk, like the stem, rather the trunk of a tree, and something like a spore mass on the top, which consists of the reproductive structures formed by this fruiting body. In many cases, but not all, the stalk consists of dead cells. The interesting point here is how similar these things look. Curiously enough, this thing called protostelium, which is also a member of the amoebozoa, is a single cell. So, there is no question of multicellularity there, there is no question there of social behavior or coming together, unless you think of its being social with itself. So, in this protostelium, you have a single cell which recapitulates the entire morphogenetic cycle that you see in these multicellular creatures. So, there is something inside the single cell which tells it how to do this, or at least makes it capable of doing this without having many other cells around. Please remember, there is no attempt here to try and trace a link in terms of serial continuity between these structures. These are all independent structures, but it tells you what such things are capable of. Pictures, you have seen some of them before, of the diverse kinds of things that you can see in dictyostelid fruiting bodies, the cellular slime mold fruiting bodies, which you can, as I say, find everywhere in the soil. I have yet to come across any kind of source where it is not found, and it is also very commonly, it occurs very commonly in animal dung, almost any animal dung that you look at. So much for similarities. What about differences? Well, there are differences of detail which are also interesting. One point of interest is that the way these things come together, so the morphology that they display when they come together can vary a lot, and you can see differences in these sketches already. That first thing is the dictyostelium discoideum type, which you have seen already, which is things coming together in the form of some kind of a star shaped mass, everything piling on top of each other. Then you get this worm like creature which crawls away and gives a protein body. Then you have this thing called, the names don't matter really, but anyway, violaceum type, where this coming together in this star shaped form is the same, but having come together, each of the arms of this star break up into a number of smaller masses, which now segregate from the others. Each of them form the miniature slug, and you get as many fruiting bodies as there are breakups. Then you have a third type in which you can see more what looks like a cloud which gathers together, not very unlike what you might think of as diffusion limited aggregation, and having come together with no starriness, with no breakup in the azimuthal direction, forms this mass, and again that slug and moves up. Finally, you have this kind of a thing called a microsporum type, where you have the same sort of cloud condensation sort of thing, mini condensates, but then instead of everything coming together, those mini condensates themselves break up into. So, my point is not to try and tell you that there are these differences, that is interesting in itself, but to tell you that they all seem to be differences imposed on a common theme, and on the face of it one would like to think that there must be some sort of common explanation for all of them, which by minor tweaking of whatever the parameters of your explanation are, might be able to take you from one sort of morphology to the other. Sorry? Yeah.
Understand. So you're saying that it follows the third row up till the second picture, and then there's a spontaneous fragmentation.
That's okay. Yeah.
Okay. So they don't actually form a slug at all.
They do. These things later, sorry, where are we? These things which you see as mounts, small mounts, they do form slug-like creatures.
Okay.
Yeah. By the way, I won't go into too much detail, but the slug itself, it turns out, is a facultative structure. So if the temperature is high, if the humidity is low, then the slug doesn't form. These aggregates immediately give rise to a fruiting body. So right. You can also find differences in the morphologies of the fruiting body. I'll say this now and I'll repeat this later. What is interesting both about the aggregation morphologies that you saw there and of the fruiting body morphologies which you see here now is that you can get these variations occasionally within your so-called standard morphology. So, this on a plate, if you have many hundreds, which you typically have very many hundreds of aggregates or very many hundreds of fruiting bodies, occasionally you will see one which look like something which it should not be, but looks like one of the morphologies corresponding to a different species. So, you do get variations on the theme, but the variations are rare and they have nothing to do with mutations, I should tell you straight away, because they occur in groups of cells and in any case they are not heritable. But these are variations found normally, and sometimes, as I said in answer to Sitabhra's question, those variations can be reproduced in the lab by tinkering with environmental conditions. Keep that in mind. Right. Diversities in shapes in fruiting bodies, lots of diversities in shapes, differences based on whether this stalk consists of cells at all. Here it does not, it is an extracellular exudate, consists of cells which are dead, consists of cells which are alive, and in some cases consists of cells which seem to be sometimes alive and sometimes dead, meaning you can get a mixture of both. So, you get variations on this thing. Please remember, all these differences that I am pointing out are traditionally held to go with species specific characteristics or group specific characteristics, as indeed they are most of the time, and many of them were used in fact for classifying these things. A small parenthetical remark here, I mentioned the number of cell types. They seem to increase not in a strict manner, but certainly in an average sense with total number of cells. So, the larger the number of cells you have, the more effective is the division of labor between these cells, and here we are speaking of the same species. So, between the same species, small celled aggregates seem to show little or no division of labor, whereas large sizes seem to show. Keep that in your mind. And across species, of course, you do get this in the average sense that I mentioned earlier. Well, there are various kinds of ways in which people have tried to confront the problem of understanding aggregation morphologies, of understanding fruiting body morphologies. So, people have tried, for example, to model mathematically the coming together of cells to see whether these mathematical models might reproduce the sorts of shapes that you see, the sorts of temporal patterns that you see. And then you do the usual thing that you do in biology, which is to try and relate the parameters of these models to things that are experimentally measurable and see whether things match up. So, there are continuum models of this kind which basically depend on diffusion of the cells themselves, diffusion of a chemical attractant made by the cells, and thirdly what is called chemotaxis, which is directed movement of cells towards that chemical attractant. Now, even verbally you can see that this kind of a thing lends itself to instability. So, if you have a local build-up of a number of cells, they serve to attract more and more cells and you get a large mass. You can also make single cell based models of the same thing, where you use as elements of your model the biochemistry of the aggregation process. You also have to throw in some movement, of course, otherwise you do not get morphologies. And the biochemistry here involves, in the case of Dictyostelium discoideum, this thing called cyclic AMP, which is the chemoattractant. So, it comes from inside the cell and is pushed outside the cell. There is something which makes cyclic AMP, serves as a substrate, ATP, and then you have this rectangular box which is the protein that senses cyclic AMP, the cyclic AMP receptor. There are other elements in this model too, most prominently an enzyme which breaks down cyclic AMP. You do not see the enzyme, but you see the arrow going away from cyclic AMP, something called a phosphodiesterase. Once you put these elements in, you put in the appropriate feedbacks, it turns out you can get very interesting biochemistry which mimics the temporal pattern of cyclic AMP oscillations that you heard of in 2 talks ago, and if you combine that with movement, you can get these beautiful spiral shapes, concentric shapes, etc., that you saw in those movies. These of course are not real cells, these are just the results of the mathematical simulation. You can also perform similar simulations to get hold of fruiting body morphologies, and here the best ones that I know of use automata type of models. So what you look at are single cells capable of being in a number of alternative states, not too many alternative states, 2 or 3 alternative states, where the states change depending on what the neighbors of those cells are, and using such models you can really get quite nice resemblances to what happens in the real situation. One thing that these models have not succeeded in doing so far, which the real situation does, is the following, which is that in many of these models, if you want to get more than one cell type at the end, in addition to getting whatever shape you are looking for, in other words, if you want to get a heterogeneous output, you need to put in heterogeneity at the beginning, whereas in real life, it turns out that you can start with a single cell, clone it, and it spontaneously develops this heterogeneity. So this is something worth thinking of. This is a greatly detailed slide that I won't go through. The point about this slide is very straightforward. I mentioned this enzyme called phosphodiesterase, or more correctly cyclic AMP phosphodiesterase, which breaks down the chemoattractant cyclic AMP. This enzyme seems to play an important role in the aggregation process, simply by breaking down the signal and therefore making it possible for cells to be sensitive to the next signal, to the second signal. The purpose of this slide is to show that when you compare the detailed dynamics of phosphodiesterase production and concentration, when you compare the detailed dynamics of these two outcomes between different strains of cells, which is to say between different genotypes of cells, which resemble each other to all outward respect, when you perform this comparison, you see that the dynamics differ drastically. So for example, there are strains in which you get a huge amount of phosphodiesterase made just at the end of feeding. Then the phosphodiesterase is inactivated, doesn't seem to play much of a role during aggregation. The other strains, on the other hand, in which this is made precisely at the time of aggregation, not much at the time of feeding. Yet other strains which you can create in the lab, which are mutated for this enzyme and don't make the enzyme at all, but they seem to lead perfectly happy lives, meaning by looking at them you can't make out that they are in any way deranged. So the point about this slide is to tell you that in addition to the sorts of variations that I showed you in the previous slides, which were naturally occurring variations, you can also get differences in detail here at the level of biochemistry and involving something which looks like an essential enzyme in the signaling process. You can get differences in detail which don't seem to matter. Now, don't seem to matter for what? Don't seem to matter for what one presumes is the ultimate, I shouldn't use the word goal, but I'll do this nonetheless to make the meaning clear. What seems to be the ultimate goal of the system, which is to produce reproductive cells to carry on into the next generation. Interesting sidelight, I told you that these cells are found in nature, everywhere in the soil as well as in animal dung. In many cases, and of course they go through their life cycles, they form these social groups in those circumstances. In many cases, these groups are clonal. You have cells consisting of the same genotype. Also in very many cases they are polyclonal, meaning they are groups of cells which are heterogeneous as regards their genotype, and the polyclonality is not small, it can go up to 7, 8, 9 or 10 genotypes. So there is a huge amount of genetic variation that you find in nature. Number of questions. and I am going to separate these questions into the developmental aspect of these social traits now. We are concentrating only on social traits, and then the evolutionary aspect of these traits. Again, detailed slide which I will not take you through. The purpose of this slide is simply to reiterate the point that I made earlier, which is that what we are talking about is not a programmed outcome gone through in the life of every amoeba of this sort, but rather something which happens based on contingent circumstances. Those contingent circumstances including things like how much food there is, what the ambient temperature is, what the ambient humidity is, perhaps the granularity of the surface, many such things, and the outcomes depending on what those conditions are can range all the way from not aggregating at all but forming a spore as a single cell, to aggregating and forming a fruiting body, to actually mating with other cells and forming this sexual structure which we have not got into at all. So, all of these outcomes are possible depending on the conditions. In any case, having come together, these cells do different things when they do aggregate, and the two broad classes of explanation for their division of labor after coming together obviously are: A, labor is divided because of certain pre-existing differences. We have already used the word heterogeneity in more than once, and these pre-existing differences, it turns out now you can show, can be in terms of genotype. So, you can mix cells of different strains and show that that gives them a bias to either form a reproductive or not. Differences in nutrition, differences it turns out in cellular calcium, and differences in other curious things like the phase of the cell cycle at which they were starved. So, I will leave it at that without getting into detail. In any case, all of these differences point to a heterogeneity existing before the multicellular stage begins, and this heterogeneity, as I said, is built upon. And as a result of that heterogeneity, one finds that within this slug there is a clear cut distinction between the front, which is going to form the future stalk, and the back, which is going to form the future spore. So, this different shading indicates that this front gives you the stalk by and large, the back here gives you the spore cells. And let me put this idea in your head, that this distinction between spore cells and stalk cells, which is a distinction between reproductive cells and so called vegetative non-reproductive cells, is exactly the same as the distinction between what we call the germ line in multicellular organisms like ourselves and the soma. So, this is a germ line soma type of distinction, but one that has come about in a different way. Now, there are experiments, and these two experiments, which again I will not take you through in detail, show that cells in the same environment can spontaneously come up with varying calcium distribution. So, this is something called a cell Salter analysis, which shows you cells with very little calcium and then cells with lots of calcium. These differences come up spontaneously in cell groups, and having come up, if you put these two together after labeling them in any way, in some way to mark them, you discover that cells which had a lot of calcium end up in the front of the slug. So, these are the same picture, and cells with very little calcium end up at the back of the slug. Again, these are the same picture. So, you can verify this guess of yours that prior heterogeneity was a factor in these things. As against this picture, and in a sense coexisting with this picture, is what is illustrated on the slide now, which is really nothing more than the example of what we call identical twins, except this is done with amoebae. What has happened here is that this slug, which would have normally given rise to this fruiting body, with the front giving rise to stalk and the back giving rise to spores, this slug is now fragmented into two. Obviously, this is a cartoon the fragmentation here is a cartoon fragmentation. Now, once this fragmentation is carried out, it turns out that this front, which normally would have consisted of cells, all of which died and gave rise to stalk, now gives rise to a miniature fruiting body of its own. And similarly, the back, which would have given rise essentially entirely to spores, now gives rise to both spores and the stalk. So, the point here being, even though there are heterogeneities which bias cell fate, and we can show that experimentally, it turns out that those heterogeneities can be overridden by social interactions should circumstances demand it. So, there is a great deal of cell-cell interactions which also goes into the picture. So, it is not that you have one or the other as an explanatory principle, but both of them are at work. These interactions, yeah, please.
Can it relate to the age of our cell, those cells? Because like if we cut the whole thing in like the first half, not first half, the first portion, these cells may have different heterogeneity in the form of ages.
Did you say age?
Yeah, age. So some are like, some of the cells are still growing, like not in that way. Some of the cells are just divided, and some of the cells are like older than those cells. Then there could be possibility that spores are the ones who are just divided, and the stalks are the one who has who is aged basically. Because you mentioned that stalk has dead cells, so there could be possibility like that.
Very good point. The actual facts are interesting. What you said is true to begin with. So, if you artificially mix cells of different ages, you can bias their fate precisely in the way that you mentioned. On the other hand, all of these experiments were done without having age differentials. So, the way the experiments are done in the lab is to starve cells simultaneously, and just to be safe, to shake them in some kind of a culture medium, so that all of them essentially are the same age since their starvation ended, and then put them together.
Okay.
So, once again you can get both kinds of things.
Right. Thank you.
Right. So, the importance of interactions is shown up in many ways. Primarily, when you measure the number of reproductive cells, meaning the number of spore cells that are made. So, this is a graph which shows interactions between two strains which are called 46A3 and 46D2. And what the graph shows is the following. You mix amoebae of these strains in varying proportions, and the proportions go from 20%, whatever, less than 20% slightly, all the way to close to 100%. These are fractions of amoebae of one of the two strains in the mixture, and what the y-axis shows is the proportion of that strain which contributes to forming spores, forming reproductives, which remember is ultimately we think the aim of coming together. And this straight line is the null hypothesis, which shows that reproductives, which makes a prediction that reproductives are formed in the proportion to which, in which these cells are present. What you actually see in this mix, however, is clearly a deficit in spore formation on the part of one of these two compounds. So clearly, at least in terms of reproductive fitness, one of these strains does worse than the other, and you can show this in terms of many other traits. The point being, so you have this heterogeneity in the wild. This heterogeneity, when tested in the lab in pairwise mixtures, does seem to have fitness dependent consequences. So, it is not a neutral heterogeneity, it is a heterogeneity which seems to matter. Same kind of thing in a slightly different experiment, which I will not go into in detail, which basically tells you that there is a big difference when you mix cells belonging to the same genotype which is clones, and there spore proportions show you something like a normal distribution, or at least a tightly bunched distribution, whereas when you mix strains which come from different genotypes, the distributions are really widely spread. I hope the point that this makes is clear. There seems to be some kind of reasonably reliable outcome when you have cells of the same genotype present, whereas you have a noisy outcome when you have cells of different genotypes present. Remember, these are lab experiments done with just 2 strains at a time. In nature, I have already told you there are up to 9 or 10 strains, which we have been unable to analyze so far in the lab. Another instance of what heterogeneity can lead to. So, these are pairwise mixes involving these 3 strains. So, you start with 3 strains isolated from nature, 3 different genotypes, you mix them 2 by 2, and in this experiment they were mixed in a 50-50 proportion, 1:1 proportion, and then you look for the same thing at the end, who contributes how much to forming reproductives, to forming spores. You see that in this mixture the ratio is biased in favor of C6, 57% to 43%. In this mixture again, very heavy bias towards C6, 76%, A3 has only 24%. In this mixture, heavy bias towards D2, approximately 70%, approximately 30%. So, you get biases, sometimes the biases are weak, sometimes the biases are very strong. But in this particular case, when all 3 were mixed in a 1:1:1 proportion, you see that the biases seem to have disappeared. So, there is some thing going on here involving cell-cell interactions obviously, which in this case has led to biases being suppressed when there are many cells. But again, the point of this picture, as of the earlier experiment, was to show that cell interactions play a big role in spite of pre-aggregation biases being important. Again, lots of experiments showing cell interactions. Now I come to something which has been thrown up by recent molecular biology work, and I may as well tell you here that molecular biology has done many things, and one quite often comes across the phrase molecular biology revolution, and this is a revolution which we are participating in and which has been running now for certainly since about 1960 onwards, well over 60 years. It's been revolutionary in many areas of biology, perhaps none more so than in the study of evolution, because it has shown us that many things that we took for granted about how evolution proceeds are actually subject to question. And the main reason it has done that is the following: using molecular biology, and here I mean specifically DNA sequencing, we can come up with reasonably objective phylogenies. In other words, with the help of DNA sequencing and some clever computational analysis, we can say with degrees of confidence that vary from case to case, but which are not bad, saying that these are true ancestor descendant links, whereas these which we thought were ancestor descendant links are in fact not so. So, this we can say. Whereas earlier the same kind of conclusion used to be reached based on what we now realize turn out to have been subjective criteria, which are perceived similarities, most often in form, in appearance. So, that has been the big role of molecular biology. And in the case of the cellular slime molds, as by the way I should say here, I will make a brief mention of this tomorrow as well. In the case of the cellular slime molds, it has overturned the conventional phylogeny, and the conventional phylogeny was, look at the form of aggregation, I showed you some pictures, look at the form of the fruiting body, decide by looking at it what looks simple and what looks complex, and work on the basis of the hypothesis, which has, we think, served us reasonably well in the past. Work on the basis of the hypothesis that evolution has taken you from simple to complex. This in short is how traditional phylogenies have gone about it. Then of course you go into more detail, see who is more similar to whom than to someone else and so on, and you start bunching things. That has been overturned by DNA sequence analysis, more precisely what's called small subunit RNA encoding DNA sequence analysis, which has shown us that these slime molds, the cellular slime molds of which dictyostelium discoideum is a part, fall in different groups, or different clades to be technical, which is ancestor-descendant relationships. These are just cartoon sketches of the fruiting bodies they form, different clades which really do not overlap at all with traditional clades. And the consequences of that for development have been many. Let me show a couple of those. So, for example, as I said, it used to be taken for granted that development involves a playing out of a genetic program. There is a deterministic picture, at least in our minds, about gene action and development. Therefore, it was believed that given that development in these organisms is separated into two distinct phases, one in which cells are by themselves feeding, growing, dividing, the second in which cells actually interact with each other, form a social group. It was assumed that this must mean that there are specific genes whose evolution in the course of their past ancestry led to single amoebae becoming social, led to single amoebae behaving as multicellular groups, as collective groups. So, this led to the idea of so-called development specific genes, meaning genes which were responsible for the social phase of development, but were not responsible for vegetative growth. And what seemed to add strength to this idea was that one could isolate mutants in which feeding was normal, growth was normal, cell division was normal, everything seemed to proceed normally, but the cells wouldn't develop, they wouldn't come together. So, you could maintain these cells forever so long as you gave them food, but if you wanted to take them through the normal life cycle, i.e. making these reproductive spores, you wouldn't succeed because they didn't work. So, it was believed that there was a subset of genes called developmentally essential genes. Similarly, there was a subset of genes, it was believed, called multicellularity genes, and these were believed to be genes whose expression was specifically enhanced after cells came together. Makes sense, right? So, you have a number of genes being expressed during the growth phase, during the feeding phase, but then when cells come together, you have a whole lot of burst of new gene expression. So, you say yes, these are involved with multicellularity. Turns out now that if you, so let me take you through this slide because the message is important. So, this work involved 385 so-called developmentally essential genes, which were defined by the criterion that their absence led to the absence of multicellular development, growth was unaffected, what I said a moment ago. Now, of these 385 developmentally essential genes, it turns out that 80% are found in unicellular relatives of these amoebae. So, these are not dictyostelium at all, these are amoebae are found all over the place, soil amoebae are ubiquitous. So, these are amoebae which are born as single cells, die as single cells, they do not know anything except living as single cells, nothing to do with multicellular development at all, and the genes are there, the DNA sequences are there. 72% of them are not specific to dictyostelium at all, you find them even in other of these, other major groups other than amoebozoa. Most curiously, if you believed that the differences among these dictyostelium species that you saw a while ago depended on the detailed working out of whatever it was that gives rise to different shapes and forms, if you had that kind of a belief, this comes as the biggest surprise, which is that a great many of these so-called developmentally essential genes, as many as 37, are not even common to all 4 of the species that were tested, which means you have two members of the same family, same broad family, in one of whom this gene seems to be essential for collective behavior, and that gene is not even present in some other member. So, clearly there is a fair amount of mismatch here between genetic makeup and behavior, and the same kind of thing, without going into detail about these so called multicellularity genes which are specifically activated during the multicellular phase. Again, you do not get overlaps across species with these very different characters in these genes. And remember, the genes are supposed to underlie something as general as multicellularity, not the details of what happens. So, where does this, what sort of conceptual scheme does this help us in building? Remember, there were two things we needed to incorporate. One was a spontaneous origin of heterogeneity, and the other was intercellular interactions, and we saw that we couldn't do without either of these two, because the spontaneous heterogeneity is there and can be shown to have functional consequences. We saw that experimentally. Secondly, we saw that if you obliterate that heterogeneity, cells can nonetheless come together and by means of interacting with each other recreate whatever it is they need to go through the normal cycle. So, you need to bring in both. It turns out that purely conceptually one can think of a starved amoeba. Starvation, remember, is a thing that sets everything off here. If there is no starvation, if there is food provided, these things go on forever, keep growing and dividing forever. So, starved amoeba, it appears, can spontaneously form either a presumptive stalk cell, pre-stalk for short. A starved amoeba can form either a presumptive stalk cell, which eventually will go on to form an actual dead component of the dead stalk, or it can form a presumptive spore cell, pre-spore cell, which will give rise to a spore. This seems to be one component of the model, but the other component of the model is that these two cell types must be capable of influencing each other. So, there must be signals going on between these two stalk cells, and between a stalk cell and a spore cell, for that matter probably between two presumptive spore cells or presumptive stalk cells, which either stabilize their existing state or cause one of them to switch to the other, depending on what the nature of the interactions is. And I hope you will see the formal you will have recognized the formal similarity between this and the sort of picture that Dr. Bose was building up a while earlier. Is there any experimental basis for thinking that this kind of conceptual scheme might be useful? Well, there is, and that comes about by comparing patterns of gene expression between these three types of cellular slime molds. This is something called acetostelium, dictyostelium in B, polyspondylium in C. Turns out that acetostelium does not form a dead stalk at all. In fact, the only stalk it forms is an extracellular exudate. I had shown you a picture of that. So, this is some polysaccharide which cells release, and they there is a negative fountain effect. They release this polysaccharide which goes and sticks to the ground, and because of the pressure that generates, the rest of the cell mass goes up. In them, it turns out that all cells are pre-spore cells and make genes, which is to say transcribe genes which are characteristic of the pre-spore phenotype, and eventually they form spores. The stalk is produced by means of material which is synthesized, these glycoproteins which are made and synthesized within this presumptive stalk cell and then later released outside. So, you have one pattern of gene expression, but within that pattern of gene expression something made which you could associate with a different pattern of gene expression, but actually turns out to be something extracellular, not a cellular thing at all. Then you have Dictyostelium discoideum, where you very early have very clear differences in patterns of gene expression between pre-spore cells which give rise to form spores and pre-stalk cells which give rise to form stalk. But remember that it is in this species that we have the best evidence for intercellular interactions influencing these two. Finally, and most interestingly, you have what is called the pallidum type, where once again you have this difference in gene expression patterns between pre-spore and pre-stalk cells, but some of the genes being some of the cells expressing pre-spore specific genes turn out to switch and go through a process called trans differentiation and become pre-stalk cells. So you start out with all cells forming the pre-spore type of pattern, but some of them now switching and giving rise to the pre-stalk pattern. So once again showing that these two patterns are not as inconsistent with each other as they may seem to be when you think of different cell types. Switching completely now. Yeah, sorry.
Can you go to the previous slide? So I read somewhere that gene always want to propagate itself to the next generation. So there are two type of different cells. One is stalk cells and other is spore cells. So we know stalk cells will not able to propagate its gene to next generation because it is sacrificing itself for being a stalk. And if we look into the different type of differentiation here, the first is each and every cells are transformed into spores, but in the second half of them, some of are only transformed into the spores and some into stalk. So I have a doubt, is there any there is high possibility then the first mechanism will be preferred by many organisms rather than the second one, because each and every cells are able to propagate itself rather than some of them. So
well, that's a deep question and one that a great many people have broken their heads over. I don't have a simple answer. I'll take you back to the germline soma analogy in our body.
Yeah.
So 99 point I don't know what percentage of the cells in our body are doomed. Doomed in the sense that they cannot reproduce. They cannot transmit their genes to succeeding generations. It's only a very tiny fraction of cells, those that we call germ cells, which are capable of doing this. Right?
Yeah.
So how do you explain the existence of this sort of seemingly stable multicellular cooperating unit, namely us?
Yeah.
Okay. Now that wasn't meant to be facetious, but that is a serious question. So very briefly I'll tell you one answer which has been given, which is that in creatures like us, we are clones. So the genes that are present in our germ cells are the very same genes that are present in our somatic cells. So even if the cells in your eye or kidneys or liver or blood or bone cannot reproduce, they can use germ cells as vehicles to transmit their own genes. That is one picture. I won't go into it in further detail now, one can expand that more. The second possibility, however, is, I shouldn't say the second, a second possibility, however, is that when you have a bunch of cells coming together, some of them reproduce and some do not. At the genetic level, it may be that a certain bunch of genes looks upon reproduction as a probabilistic phenomenon. And now the game shifts. So the gene's eye view of this could be the following. So if you have two individuals A and B coming together, in one situation A might reproduce more effectively than B, but in a different situation B might reproduce more effectively than A. So on average, in the meta population at large, the two may be on a par relative to each other. You see what I am trying to say?
Yeah.
So, in any given single fruiting body, this is true. Some cells invariably lose out in this genetic race. If those cells happen to have a different genotype than those that reproduce, it may be that they have lost the race, but only that time. There may be other combinations in which they come up ahead. Now, is that a satisfactory answer or no?
Yeah. Got it.
Okay, I am not satisfied myself, but that's one of the things that one
Wait,
can
yeah.
think of. Okay, so as I said, now I switch completely to this analogy between multicellular development and social behavior, and the aspect of the analogy that I wish to consider is precisely the one that you brought up just now, which has to do with division of labor, and it has to do with looking at social behavior from two viewpoints. One is what you might call an economic viewpoint, productivity viewpoint, GDP viewpoint, which is very popular in our country now, and the second from the purely reproductive viewpoint. And you can just as well use either of these criteria as the ways of assessing social groups. So, if you look at a honeybee colony or an ant bee colony, you can ask many questions about it, but certainly two questions you can ask are: one, how productive is the colony in terms of preparing honeybees or ants for the next generation? So, how well does this nest do in terms of seeding other nests? And the second question you can ask is, within this nest per se, how happy and contented are these ants and honeybees? Do they have enough sugar? Do they have enough nectar? Do they have a clean colony? Are they rid of all fungal pests, etc., etc.? Economic versus reproductive. Now, let me point out a couple of phenomena. So, these are experiments, lab experiments I should say, done with queens of an ant, something called a harvester ant, which is known to exist in the form of a solitary type and a cooperative type. So, in nature, you have harvester ants which are entirely solitary, you have harvester ants which are entirely cooperative. Important point to notice is they all belong to the same species. So, in some way these are like cultural differences, these are not genetic differences, but the culture is tied to whatever group you are looking at. There may be deeper genetic differences which have not been discovered so far, but don't appear to be there. So, the experiments which were done here was to set up synthetic groups in which you had solitary H and, so H is the symbol for solitary here. I am sorry, this should have been Q, or that should have been P, whichever you want. Anyway, solitary ants belonging to the cooperative group, pair of cooperatives, pair of solitaries, and a combination of one solitary and one cooperative. I hope the picture is clear. So, what is being set up here is artificial ant groups consisting respectively of single ants which are either by origin solitary or cooperative, or pairs of ants which are either both solitary or both cooperative, or one cooperative and one solitary. As you might expect, oh, by the way, yeah. So, what is being counted here is the reproductive efficiency of these groupings, which is a measure of that is taken here as the number of eggs produced by this grouping. Now, as you might guess straight away, pairs of ants in general do better than single ants. Okay. However, there seems to be no difference between the egg. I am thinking of these here. So, this as a group does better than this as a group. However, so it's always better to be with someone in order to run a functioning nest from the point of view of reproduction. However, within these three groups, there are no significant differences between HH, PP and HP. So, whatever the supposed inherent proclivities of these ants, once they come together with another ant and decide they have to live together with another ant, they function in the same way as far as their reproductive efficiency goes, as far as the number of eggs produced by them goes. So, I just point that out as one thing. So, their social dynamics decides what is going to happen, not their ancestry, so to speak. Yes, please.
Size, can we see the differences then?
I'm sorry, please, I'm sorry, say it again.
So the group, even when you made a group in the pairs, there were only 2 ants. So isn't the group size really small? Suppose we make the group size really large. So would the conclusions be different?
I can't answer that because the experiment was done by someone else. But the point is that in nature they seem to begin, the cooperative one seems to begin in pairs. So this is just an attempt to mimic the kind of thing that happens.
Sure, but isn't the group size really small to make these
No, the
conclusions?
group size grows eventually. Do you mean the number of replicates? The number of replicates was large enough, that's not shown on this slide. You mean the number of times the experiment was repeated?
No, the group size. So suppose we make the group size from 2 to 7 or 8. So you don't have.
So would this saturate at large starting sizes? Honestly, I can't answer. But in nature, they seem to begin in pairs, and this was what the workers were trying to imitate here. Okay. Now, an experiment which asks a similar kind of question, again in the harvester ant. What it does here is to look at one of the situations we saw a while earlier, which is to set up pairs, but in this case pairs of solitary ants. So, ants which are supposed to be solitary, which normally never form pairs, but are being put together here. And once again, what is being measured here is the number of offspring that result here per capita, please notice, so per head. The control is where you have only a single ant, which is what normally happens, and these are the number of offspring on average that result, something around 4. On the other hand, when you have pairs of ants, the per capita thing comes down just a little bit. Obviously, the productivity as a whole increases a lot because there are 2 of them here, not 1 anymore. But the interesting point is the following. The interesting thing is that among these 2, beg pardon, among the 2 that form this pair, who is the egg laying member, who is the one who actually reproduces, varies from case to case. This is point here, so let me repeat. So these are solitary females of 2 different origins, think of them as A and B. These solitary females are brought together, forced to live in pairs. They do better at reproducing as a pair than they do individually. But the one among the pair who reproduces differs from case to case. The one who reproduces is usually called the queen, the other one simply is a worker. So this perhaps comes somewhat closer to the question you were asking. The one who is reproducing in terms of ancestry differs. So one of them clearly has no direct genetic benefit because she does not, these are all females by the way, she does not have any reproductive advantage at all of coming together. However, that does not mean that that is invariably the case. So there is no fixed heterogeneity between these 2 ancestries that makes one more capable of reproducing than the other. Slightly more complicated slide, but an experiment done this time with paper wasps, which are also social insects. Wasps are also social insects, with the same kind of question in mind. And here, actually, this is somewhat more interesting. What's being looked at is both productivity in terms of number of young eggs produced, as well as division of labor. And of course, when you say productivity and you say young produced, you are asking, implicitly asking who is laying the eggs, and it's invariably one queen. Turns out that what is interesting here is that when you combine a pair of these paper wasps, by the way, paper wasps are naturally social, so this is not exactly the same situation as those ants, but when you combine a pair of these paper wasps, one of them becomes a queen, the other becomes a worker. That much is granted. So there's some kind of Q or asymmetry that they pick up. So there is reproductive division of labor. So they differ in this sense. However, there is no somatic division of labor. What does somatic division of labor mean? So that means in this case, things like cleaning the nest, guarding against predators, going out and getting food, foraging, this is shared equally between each of these two females. However, one of them retains the right to reproduce, the other one does not. So there is some asymmetry, but no, not a different kind of asymmetry. What is interesting is that when this number, and this perhaps addresses the sort of question that the gentleman there was asking, if this number is now increased to 3, again you have social behavior, again you have reproductive division of labor that you invariably have. So one of them is the queen and the other two are not queens. But now, in addition, you also get, it turns out, somatic division of labor. So between these two who don't reproduce in any case, one of them is a forager and the other one takes care of feeding larvae eventually, that sort of thing. So you get a more complex kind of division of labor, which in the multicellular case you would call differentiation into different cell types once you have larger numbers. So the point of all of this is not to get into the details, but basically to tell you that there are situations paralleling what we saw in the multicellular development case, where large numbers per se, and therefore the increased complexity of interactions per se, seems to result in outcomes which therefore are not simply based on an inbuilt genetic program. They cannot be, because they are so strongly influenced by contingencies. Yeah.
Result is interesting, but I don't know how easy it is to perform the experiment. What if one could have kept on increasing the number? Would you have more and more division of labor and eventually sort of saturating?
That to my knowledge has been done, by the way. I'm not sure whether it's been done with this paper wasp, but it has been done with a honeybee, and what you say happens, yes.
So it sort of saturates eventually?
Beg your pardon?
Does it saturate the division of labor?
Saturate meaning you get the, okay. So the parallel being made here, the analogy being drawn here is between number of cell types in a multicellular organism and the number of behavioral types, which are in the literature called castes, in a social insect. And so there is a certain number of task specific behaviors known. Yes, it does saturate. Yeah.
Thanks.
Those of you who are interested, especially I find this overlap between social dynamics and biology fascinating. There's a nice book by Oster and Wilson with details of precisely that kind of experiment involving modeling too. Okay. Pardon?
The per capita productivity is dropping with the
first in the you're looking at the right hand side, right?
Yeah, right hand plot.
Yeah. So so this is a single female by herself. This productivity on whatever the scale is 30 and something. Let's say 35, right? Here per capita it's about 15, so becomes 20, and here it again becomes well. Here it rises much more. Here it's probably 45, right? If you take that as 15. So in this picture, 3 wasps seem to do much better in terms of reproductive efficiency of the nest than a single wasp. Presumably drops with 2 wasps simply because of the time being spent on this non-reproductive tasks. So you don't have any specialist here to take care of those tasks.
So how you're defining productivity over here?
Usually number of eggs laid, I'm not sure
Okay.
whether they in this experiment looked at how many hatched, but usually in terms of number of eggs or number of larvae.
Okay. So,
So
alright.
here your arithmetic would say, let's call that 35. So 35 into 1 is 35. Here what is that? Maybe 18 into 2 is 36 there. And here you have 15 into 3, which is 45. Should be, yeah. Yep. Yep. Is that okay? Right. So I'm essentially ending now. And the question now comes, taking this very, very broad view, there are cues on which individual cells in one case, individual organisms in the other case, there are cues on which individuals build to interact with each other. These cues in some way seem to be part of their background makeup, they are inherent in some sense. So, unlike in experimental situations, in nature, as far as we know, there are no forced differences between individuals in these situations. The question comes about what sorts of cues could they be? Well, one candidate which used to be extremely popular in the history of developmental biology and in the history of evolutionary theory, one candidate is nutrition, which can translate into many different things. Let's say how big you are, how much food you have eaten. It has to get a bit more sophisticated, probably also the quality of food you have eaten and so forth. So, nutrition related cues which translate into metabolic state more generally seem to be good naturally occurring candidates for the sorts of spontaneous heterogeneities that can arise between individuals, which would feed into their probabilities of reproduction. Okay. And there is a rather nice experiment done very recently by Sunil Lakshman and his group, where they looked at a clone of yeast cells growing on some kind of agar containing nutrient medium. In one situation with high concentration of glucose in the agar, so these cells thrive pretty well under these circumstances, and you see a growing colony. There may be I don't know, perhaps 1 million cells within that colony which moves out. There was a picture of this colony growing somewhat earlier, which was rather nice. I think Bhaskar showed one such film, but that was in Dictyos Chilem, where you had a colony growing out in the form of what you could imagine was a circular wave, but inside the colony there was all kinds of nice things going on, finally giving rise to fruiting bodies. There are no fruiting bodies here, though yeast in other situations does give fruiting bodies, so it is a growing colony. Same kind of colony, except that this is in low glucose conditions which are limiting. And the point here of course is that competition between cells becomes important. Why? Because the nutrition levels are not, the potential amount of nutrition in the environment is pretty low. Interestingly and fascinatingly, it turns out that under these circumstances, these colonies throw up incredible patterns, spatial patterns. Underlying these spatial patterns are interesting temporal patterns which I won't go into here at all, but very interesting spatial patterns. And to the eye, these patterns look like a difference between light cells and dark cells. The dark cells are especially visible there, and here, somewhere. At least you can see the growth pattern very easily. It's a opening rosette kind of thing. What is interesting is that these patterns seem to discriminate between two types of cell which have arisen spontaneously here. So this is really an example of differentiation, and this is occurring in yeast, which has no business showing anything which is multicellular like, as far as we have known and as long as we have known, an obligatorily unicellular organism. But this differentiation consists of dark cells storing a lot of glycogen So carrying out what's called neogenesis with the help of that stored glycogen, and dark cells also at the same time storing up a lot of trehalose as an energy source. So basically dark cells have lot of trehalose, light cells don't have any, dark cells have lots of glycogen, light cells don't have any. So this is a clear-cut difference in patterns of gene expression, what you in other situations would call differentiation, which has arisen spontaneously here, based presumably on slight differences in metabolic makeup between individual cells. So clearly, combined with these slight differences, you have to include intercellular interactions involving cross feedbacks as well as presumably self feedbacks, both positive and negative cross feedbacks, which stabilize the pattern. Now that still doesn't tell you why you get this spatial ordering. That's a different story altogether, but tells you why you get this sort of difference. So that's my last slide, trying to tie up these two themes of development and multicellularity based on the small example of the cellular slime molds. So how might multicellularity have evolved? And one possibility which these observations throw up is that it could have evolved as the spontaneous outcome of self-organization in a very general sense, and then division of labor which has come up simply based on inherent pre-existing properties among these cells, and those properties might be nothing more than intercellular interactions and feedbacks without any genetic change yet. So go back to the yeast cell example that I showed you a while ago. There are no genetic differences here, but there is very clear-cut division of labor. So without any genetic changes, these spontaneous differences might have arisen and may have become stabilized later, and this is a theme that I will try and pick up tomorrow, may have become stabilized later by means of secondary genetic changes. So the picture here is not so much of natural selections for genes which, so to speak, do something new or result in something new being done, as natural selections for genes which stabilize the difference which pre-existed their coming into place. And this is now, when I say now, since quite a few years, now becoming more and more spoken of as a useful way of looking not just at development but also evolution. And the central focus of this point of view, of course, as you can see, is that the change, the phenotypic change, precedes the genetic change, at least the genetic change in question here. So I will end by leaving that thought in your mind, and we will try and take up this sort of view of evolution tomorrow. Thank you.
Questions? Yeah, Rahul.
More of a comment, but yeah, about your slide showing, for example, the Sunil's yeast work, and I mean, so in all these cases, whether it's, I mean, the Dictyostelium things could be clonal colonies, though they tend to be heterogeneous. The yeast was clonal, and of course we are basically clonal, multicellular. So my question, so it seems reasonable that even in Dictyostelium, the genetics has evolved to stabilize this kind of multi-function body, and that could, yes, have given rise to multicellular organisms. So I think my question is more like, is there any sort of missing link between slime mold kind of creatures, which are, they look like worms, I forget what you call them, those things, and
Slugs.
and actual things like C. elegans, which are also multicellular, they look like slugs, but have this internal morphology and all that, which slime mold doesn't. Is there anything in the middle that we can say somehow this evolved into what we know?
Okay. The slime mold pattern of making a living and our pattern of making a living differ in one essential respect, of course, which is that we resulted from single cells which became many cells via the products of cell division remaining stuck, whereas these involve single cells becoming many cells via many previously free living cells coming together. So this seems to mark a fundamental division in origins between the two. So there is no missing link or any kind of link between them, and as I said, the only common root of all of these lifestyles seems to take you back to a single celled ancestor. It doesn't have. So the aim here is not trying to say how one could have led to the other, but rather to say that single cells came pre-equipped with properties that enabled them to make a go of it when they found themselves in multicellular groups, where it would have been advantageous to live as a multicellular group with the enhanced productivity that accrues thereby, etc., etc. So they found it possible to live that sort of a life, or more properly to go through that sort of a life cycle on the basis of properties that pre-existed. Okay. But now the problem was that those outcomes were strongly dependent on contingencies. Or to put it in different words, they were noisy outcomes. Genes, the genes of the sort that we have in mind now, came into play to improve the signal to noise ratio to a stage where the outcome is now essentially 100% assured in some lineages. So the advantage of looking at the slime molds and similar creatures is to see that there is some living on the border where both this and that are possible, and then to ask what makes transitions in either direction more likely.
So if I may just comment, actually, so this is a theme that came up after Archishman Raju's talk, so where he was basically showing how the epigenetic landscape of Waddington could be seen as basically suggesting that some of the furrows were there, but they were shallow and not explored by development earlier, but because environment changes, as a result they would become deeper, and we would see the cells emerging with different phenotypes, and we would exclaim that, oh, it's like a genetic change, but they were pre-existing. It's just that they were not explored because they were much shallower than the deeper furrows. So essentially I thought this nicely relates to this theme that has often been mentioned recently, that co-expression might lead to co-regulation. So like what you said, that noise could lead to co-expression, but that subsequently leads to co-regulation.
Purely at the level of first, before that, you'll see a repetition of this theme of the epigenetic landscape tomorrow when I speak. So that's one thing. But the second point, if you, like me, like analogies, which by the way are believed to be bad for your health if you believe most people, but if you like analogies, there's something very interesting between what you said just now, co-expression leading to co-regulation, and what is known to be a feature of synaptic learning.
Yes.
So this is really the Hebbian synapse in biochemical terms.
Absolutely. Yeah. Any other questions or comments? Okay. Yeah, sure.
This experiment that you showed, whereby changing the nutrition composition, you have the ratio of light cells and dark cells changing. So, but that doesn't show the emergence of new cell types, though. Right? You're just changing the balance.
Yeah, I mean, what do you call Again, this is a theme that I think I partly touched on when I showed you that. Oops. I partly touched on in this slide, which is there is a transition in the course of going from one cell to many cells, or in the course of going from one, I don't know, harvester ant or wasp or honeybee to many ants or wasps or honeybees. There is a transition between the same cell doing different things at different times, right, to many cells doing different things at the same time, right. Now, would you call that differentiation? That's a point of terminology. Would you call that two different cell types? You would certainly call them two different behavioral types in the case of organisms. And what is nice with some of these social organisms is that these behavioral types also go with very strong differences in morphology. By the way, for those of you who are interested, among the many questions that bothered Darwin about social living, in particular the social insects, one bothered him a lot. Of course, the first problem is to figure out why only some individuals reproduce and others do not. Your question, why only the queen reproduces and others do not? What's in it for them? Why are they doing this? So that's a big question. Second question, which in some ways is even crazier, is that in many of these cases, the individuals that do not reproduce, now bear with me, the individuals that do not reproduce in many different ways, right. So in other words, you have many different behavioral types or behavioral castes, which are also morphologically very distinct. With ants, this is well known. So you have major soldiers, minor soldiers, and you have that with other insects too. You have a clear-cut distinction between foragers and larval feeders. Okay, so I think the parallels run deep. In the case of the yeast cells, you can say, yeah, they look the same. Of course, they look the same, except for that light-dark color, which essentially is a cell density-related artifact and an optical artifact. But that apart, very clear differences in patterns of gene expression. Some are doing glycolysis, others are doing gluconeogenesis.
All right, so on that note, let's thank Professor Nanjudaya once more. So we have no formal group sessions, but tea and coffee has been arranged at the 4th floor lounge so that we can have a discussion regarding the presentations tomorrow. And Shakti has a few words to say about these group project presentations.
Okay, so as mentioned, you will be giving your talks tomorrow throughout the day. We were initially sort of having a bit of a debate as to how we should go about ordering these talks. At the end, we sort of decided that we'll just go in the same order that the groups were announced. So I mean, most of you would know your number, group 1, group 2, and so on. If you don't know, please come to us and we'll let you know. But in addition, if any of you have some constraints, like if you need to leave by lunchtime or something like that, or you like to talk early or like to talk a bit later, please let us know. We can try to move you around. But barring that, we will just go in the order that we had discussed, so that we had assigned you on the first day. Now, I'm not sure how many of you have been checking out the website. We have been updating the website throughout the workshop. So right at the top, we have next to the schedule, we put a new link. We had put a new link. It's been up for a while now, where you can get all the details of the group project. So this is actually just the same document that I read from on the first day. Right at the very end, I had mentioned. Oh, okay, right. Let's take this mic up. Yeah, I mean, I think by now, a lot of this, is not so relevant because presumably all of you have finished working on the projects by now. But the only thing that matters really is the presentation, right? So initially we had planned it for like around 20 minutes. So I think we will probably stick to this. We might extend this to 25. We still not completely sure, but we'll see how it goes tomorrow. See how much. Based on the time that we think we have. But this is roughly how we've planned it. And as I had mentioned before, there's no sort of requirement that each person speaks the same amount of time. I mean, so there are groups of 3. So we had hoped that everybody is going to contribute equally to the group, or at least in their own way, right? Everyone has a different background, different experience, and so on. But you don't all have to speak for the same amount of time. If there's somebody who wants to speak for a bit longer, someone wants to speak a bit less, that's fine. We would like it if everybody speaks a little bit, but it's up to you. Also, in terms of how you want to present the talk, you can present it in the manner that you wish. But the only thing that we would prefer is that you at least mention these 4 things. Basically, you talk about the question, the biological question that drove the project. That's really important. You talk a little bit about the rationale behind the choice of modeling approach that you've used. Why exactly did you think that this particular way of modeling would be a good way to answer this question? What have you observed in your simulations? Of course, this is a must. And what does this mean? Like do the results tend to support what's seen in experiments? Do they contradict it somehow? If so, is there some assumptions that maybe one needs to take into account? Could this lead to some prediction? I mean, there's a whole bunch of things that one can infer from these results, right? So that's about it. So if you can just cover at least these four points during your talk, which would be around 20 minutes or so. So 15 minutes plus 5 minutes of questions. But like I said, we could possibly go up to 25 as well. So yeah, so that's basically it. Does anybody have any questions regarding the talks tomorrow? Or if not, we will head over to the coffee lounge. Yeah, if you want to ask us any questions individually, we'll be up there. Okay, so if there are no questions for the moment, let's head over to the coffee lounge. Thanks.